Thanks very much. It's an honor to speak to you today, and yeah, for the packed crowd here, there's some seats up in front of there. For those of you that are joining us online, really happy to be able to tell you about a project we've been working on for the last several years, and in addition to myself and Maxim, Maxim in particular has really been driving this work. We also have a great collaboration with Anshul Kundaji in genetics and computer science, Rob Tibshirani, and also co-first author Aaron Clark of one of the papers we're going to talk about. So looking forward to this discussion. We'll show some slides. And then for the last part of the presentation, we'll have a nice discussion with Dr. Bhatt and take questions from the audience about this. So we're talking about applying machine learning methods to the antibody and T cell receptor genes as a way to try to identify what's happening in a immunological disorder or other sort of medical condition for people. And we should just mention disclosures. So I've done, consulting with some companies, and none of that's related to this talk. And also some of the work that or IP derived from this paper that we're going to mention is the basis of a startup company that's beginning now. So just for full disclosure, but we're not going to talk about anything related to the company in this presentation. So as many of us have witnessed, sequencing and other sort of detection of RNA and DNA sequences has become a bigger and bigger part of medicine, and really important for diagnosis in lots of different disease areas. Everything from detection of cancer gene mutations, identifying the basis for genetic syndromes, rare diseases, and disease risk more broadly in the population, and also in infectious diseases, for example, pathogen detection is a useful way there. But there's a sort of niche area of genetics in humans and other vertebrates that is full of information, but we haven't really made that much use of it previously in diagnostic practice. So these are the genes that encode immunoglobulin receptors on B cells and T cell receptors on T cells. And the really interesting part about those parts of the genome is, unlike every other gene type in the body in people, these ones they're made by genetic rearrangements that happen in the germ, that happen as new B cells and T cells are being formed. So the actual germline genome is being reassembled, stitched together in sort of randomized ways for the seemingly evolutionary purpose of generating diverse receptors that recognize almost any antigen you might come across in life. So the immune system strategy selected by evolution seems to have been generate massive amounts of diversity in these receptors. And then when you need a particular response, by chance, you're likely to have at least some cells that will recognize it in the body. So, so by their very nature, these genes actually have a lot of diversity, a lot of potential explaining of what's being reacted to in a person's immune system. But it's a complex kind of sequence data. So that's what we're going to get into today. And there's one area where this has been used a lot, which is if somebody has a lymphoma or a cancer derived from B cells or T cells, these kind of gene rearrangements that encode antibody or T cell receptors can be a really good unique marker of that particular malignant cell clone that might be the cause of the person's cancer. So just to say that the other aspect of these genetic sequences that's really useful is that they change over time. Most of our genomes they're locked in, except for occasional somatic mutations that happen throughout life. But the B cell and T cell receptor populations, they reflect which B cells and T cells are in the blood or in the tissue that you're examining at a particular point in time. So they're dynamic and they can change according to the person's health state. When there's a response to a particular antigen, the B cell and T cell clones that respond to it, they undergo cell division, they expand their numbers tremendously. So it gives a big signal that you can look for in somebody's immune system. So we wanted to see whether we could use those as a way to interpret what's happening in a person's body and maybe diagnose what either immune response they're mounting to an infectious agent, or what kind of autoimmune disease they may have, or other sort of immunological response. So as I mentioned, we have this random recombination, clonal expansion when the cells respond to something. And then there's also other features that are useful for interpreting what the immune system is doing. In the case of antibodies, they also accumulate additional mutations in the genes that are encoding the specificity determining part of the antibody. Those are called somatic hypermutations. We can also track those. And antibodies can also switch what constant region they're making, which is the stock of the antibody, and that can determine the function of the antibodies that are being produced. So those also have like sort of functional information that we can read from DNA sequences, importantly. So with the great diversity of B cell and T cell receptor repertoires, everybody has, a huge universe of receptors that they may be the only ones that have that particular exact rearrangement. But if we look at people responding to the same antigens, we do occasionally find some receptors that actually have very similar sequences. Just the selection for binding actually draws out of the vast pool of diverse receptor a particular pattern that you can detect. So that's something that we've seen it in previous work. They're still relatively rare. So if you look at two people getting the flu vaccine, for example, there might be only a few percent of all the B cell sequences, B cell receptor sequences that look similar between any two people. So most of your response seems to be sort of private, but there are these public ones that can actually tell you what the person's responding to. And this has been less explored in areas outside of infectious disease or vaccination, but we think that those patterns are there as well. So there are some other things you can notice, for example, like during the COVID pandemic, we saw that a really characteristic finding of people's immune responses was that if you look at all the B cell clones that are in somebody's body, and you just look at what the somatic mutation state is. So here we have green being very little mutation, which we see in the IgM and IgD sequences of B cells in the blood, because they're naive cells that have never encountered any antigen, versus ones that have class switched to other isotypes like IgG or IgA subtypes. In the response to a new acute infection, you see a lot of B cells that have switched to a new class. They make IgG or IgA antibodies, and yet they still haven't mutated the antibodies very much. So you actually still have low mutation. That's a pretty strong signal of a new response to a new infectious agent. So that was very obvious in COVID, but also shows up in other infections. Also, we've just seen in chronic conditions like HIV, for example, infection, there can be changes in the proportion of receptors that use particular gene segments. For example, these are the variable gene segments in the heavy chain of the antibody. People with long-term HIV infection show expansion of B cells using particular receptor types. So this one is a strong signal of this V434 V gene that's in the heavy chain. So that can also give us a clue as to what somebody's responding to. And as I mentioned, in any condition, if we look at a lot of people that have had the same exposure, we eventually recognize some B cells or T cells making similar rearrangements. And that can give a clue as to what they're actually, what the activity is in the immune system. So we call those often convergent or public sequences. And IgH here just means the heavy chain of the immunoglobulin genes. So these are the kinds of things that are going to give us the signal to allow us to detect what somebody's medical condition is or immune response that's going on. So this paper that we published in February was applying machine learning methods to these complex sequence data types to see if we could classify correctly a number of different human immune states. Maxim was, I would say, the main driver of all this work. But it was a huge group effort and involved many really valued clinical collaborators and others who did everything from working on the analytical methods with us and also sharing clinical specimens from really well-characterized patient cohorts that they'd been studying. So we express our thanks to everybody involved in this. The main thing is the input data are the receptor sequences from the B cells and T cells, so the antibody heavy chain and the TCR beta chain. We then train a series of 3 different models to go into this machine learning approach that I'll describe. And then from that, we sort of combine them to give an overall prediction as to what the person's immune response or medical condition is. So the key form of data, we're basically amplifying the heavy chain genes from the B cells and the TCR betas from the T cells. And they all have this sort of structure of having a variable region, a diversity region, and a joining region that make up the part of the antibody or TCR that really determines the specificity of what it's going to bind. So this is where all the sort of information or informative aspects are really locked in. And here are the 3 models that we used in this machine learning approach. This is a really important aspect of it, and it gets into some of the subtleties of these kind of data. We picked 3 different models to look at different aspects of the immune receptor repertoires. And choosing which features you want to use the machine learning approach on is really important in this kind of thing. Like if you just feed in the whole sequences, maybe if you had enough data over time, you could actually just rediscover things that we already know from medical and immunological research. But if you actually point out for the models in advance features that are really important, we know for binding, for example, or for their other functions, it really helps the system to perform better. So that's why we have these 3 different ways of looking at the receptors. So the first one is this overall composition of the population of receptors in somebody's B cells and T cells in the blood. And I should say the place where we're actually obtaining all these data is from blood samples of the people. You could actually use it to look at tissue biopsies and things like that, but There would be other differing factors like what populations of cells are in the different tissue sites. So this is all standardized to blood. So in this first model, we're basically looking overall at broad features, like what's the proportion, what's the frequency of use of particular variable genes, what's the level of somatic mutation for the antibodies of different isotypes. Those are the sort of characteristics that go into this part of the model. So this is really like a very broad overview, but it can still be very informative. Like, for example, it's been noted in some conditions like ankylosing spondylitis, for example, autoimmune disease condition, there can really be strong overuse of particular V genes in the T cell receptors in that state. So that's actually something that, even at this very crude level, there's actually a lot of informative data. So we incorporate those basic bulk features, and those are the inputs that go into training this sub model for the overall combined model. So then the second part of it is looking at the key region called CDR3, which is the complementarity determining region 3. That's where the V, D, and J gene segments are actually fused together. It's that portion of the variable region of the whole immunoglobulin or TCR beta. That's the part that's the most diverse in sequence, and it's actually probably the most important for determining the specificity of the antibody or the T cell receptor. So it's a really key region, and it forms a loop that's part of one of the sets of 3 loops that make the main binding contacts with the antigens usually in each of these receptor types. So in this case, you can see here this region where the V, D, and J join. Those together, that junctional area kind of encodes the CDR3 part. So here we can use a simple sequence clustering or sort of comparing the sequence distance, Hamming distance between different receptors, and look for ones that are very similar between people that have the same exposures. So we do a basic clustering approach by looking at the distance of sequences, like how many mismatches there are, and we can use that as a way to try to identify similar sequences between people. That's also relatively straightforward. Then there's a third approach that we used in this, which hadn't been used as much, especially when we first started developing this methodology. In addition to looking at the real primary amino acid sequence, just the ordered amino acids that are making the chain up, we can also use other ways of representing the protein sequences. So we've all heard of language models, for example, right? The massive language models that are used to be able to power things like ChatGPT, where they've been trained on essentially all the text available in different languages that humans use. And by training models to predict what word might be the next one in a particular sentence, they can actually have amazing predictive or text generative properties, right? Here, a similar kind of scheme is being applied to protein sequences. So here we're basically training a neural net with protein sequences that are coming from, all the different kingdoms of life, but masking certain amino acids in the protein sequence. And if you train these models well enough, they can actually predict what's the most likely amino acid that's been masked in a protein sequence that you give them. So that's actually, over time, these models would learn, well, what are the most characteristic sequences, or what are common patterns? What are the certain sequences you hardly ever see in real proteins? So these sort of things, they give a very different representation of the actual protein sequence than just the string of amino acids. And they basically would convert the primary amino acid sequence into a numerical vector. And the interesting thing about these protein language models is that they actually seem to be able to learn or incorporate a lot of information that relates to the evolutionary history of the organism that the protein came from, or the function of the protein. For example, like particular domains are able to be predicted with this sort of approach. So it's a different way, one that's hard for us as humans to really intuitively understand if we just see this numerical vector produced from the protein sequence, but it does seem to encapsulate a lot of functional and evolutionary knowledge about the sequences. So it's really analogous to here. If you have a sentence, early detection of disease greatly improves blank outcomes. A normal language model for English could actually guess that patient outcomes might be a reasonable choice here. In the case of the amino acid protein language model, it's a similar thing. If you block out one of these amino acids, the model once trained can actually guess what's the most likely amino acid. And the embedding that we see is really this sort of numerical representation. So this is really good. Machine learning tools can work on that sort of space. And this is an important part of the overall model that we generated. So for example, when people have used these inputting data from all kinds of different, bacteria, mammals, other vertebrates, all kinds of creatures, you can see that from distinct regions of the kingdom of life, you can actually see that there are certain common patterns that show up in these protein language model representations. And when applied to immune system sequences like antibodies or TCRs, You can tell the receptors that come from naive B cells are these blue ones, and memory cells, these red ones. So there's some real characteristic sequence differences that show up in this representation approach. And we think it's actually giving more information than just the literal string of amino acids. So that's all part of this. So we basically embed the BCR and TCR sequences in this sort of a protein language model. And here we're using one called ESM2 that derives from the company Meta's work on this area. And we have that as part of our overall model. So we can train these things based on healthy individuals or people with different diseases. And based on that, we can develop an overall classification approach. So I think next, Maxim Zaslavsky is going to present the next part to talk about some of the details of the next steps of the model.
Thank you so much, Scott. So at this point, we've seen 3 different ways of extracting features from B or T cell receptor sequences and using them to train classification models. And the next choice we made in this project was to find a way to combine all these models, to leverage their individual strengths, but also to cancel out any of their individual weaknesses. This is a common approach in machine learning called training an ensemble model, to be able to resolve potential errors of these individual methods and put all this information together as much as possible. And that was the next step here, which was to train this final model that takes as input the predictions that these 3 B cell and 3 T cell receptor based models make and arrive at a final prediction for the patient. So the way this approach could be applied to new patients is you generate sequencing data from B and T cell receptor repertoires. You feed them into those 6 base models. You feed their outputs into this final ensemble model, and you arrive at a final prediction of disease for this patient. We call this framework MAL-ID for short. And to evaluate it, we assembled the data set with just under 600 individuals from 6 immune states. These immune states included COVID-19 patients, individuals with HIV infection, influenza vaccine recipients at day 7 after they received the vaccine, when the B cells responding to the vaccine are usually at peak frequencies, lupus patients, type 1 diabetes patients, and healthy controls. So this was intended to span a wide variety of possible immune states to see how this sort of approach could distinguish these broad categories. And as a result, we have the 2 viral infections, the 2 autoimmune diseases, a post-vaccination signature, and the controls. As far as testing this model, we took a few approaches. The first is a technique called cross-validation. This is our first way of evaluating how well the model works, and it's quite straightforward. What you do is you take your set of patients and you divide them into a training group, 2/3 of the patients, and a testing group, 1/3 of the patients. And you can do this several times using a different 2/3 and a different 1/3 each time. And every time you do this split, you retrain the models from scratch, so they never see the patients that are selected for the test set, and you record how well the model does. If you combine all those results, here's how well we can tell diseases apart in this first proof of concept study using immune data. This is called the confusion matrix. So the way to read it is the rows here, highlighted in red, represent the true diagnosis of a patient. There you have the viral infections we spoke about, the controls, the flu vaccine recipients, and the 2 autoimmune diseases at the bottom. And the columns represent the predictions. So if you look at the diagonal, which we're highlighting here in red, that's the correct answers from the model. That's when the model predictions line up with the ground truth for the patient. There are many ways you can measure performance of a model like this, but just to show you one classification metric of the many we've evaluated in the study, here is a metric called AUC. That's short for area under the receiver operating characteristic curve. The way to interpret this metric is, let's just take a simple example. Let's say you have 2 classes. You have COVID-19 patients and you have healthy controls. The AUC score is the probability that a random sick patient would have a higher score from the model than a random healthy control. So it's measuring, is the model assigning higher COVID predicted probabilities to the COVID patients compared to the healthy individuals? And this metric can range from 0.5, that's the baseline when the model isn't able to separate the classes, all the way up to 1.0 when the model is perfectly separating the classes. So what we're seeing here is if you compute that score for all pairs of classes in our data set and average that up into a final multi-class AUC score, you have 0.986 by combining these B and T cell approaches together, which is very close to that 1.0 extreme that we were talking about. So far, everything that we've discussed today has been in the context of evaluating for multiple diseases at the same time, right? A simultaneous screening starting from this blood sample data for multiple acute and chronic infections, as well as autoimmune disorders. But you can also specialize this approach to focus in on one particular condition. The example here on the right in red is focused on lupus versus the other diseases in the model as background. And the way this works is that MAL-ID gives you a predicted lupus score for each patient. And then you have to make one more choice if you're developing this sort of a model, which is what is the decision threshold that you use to determine where you draw the line for how you cut those predicted lupus probabilities. Anyone above that score would be declared to be, yes, this is likely a lupus patient. Anyone below that, the model would be predicting, no, this is probably not lupus. And depending on how you choose that threshold, you get different sensitivity and specificity values. And that's important because depending on the clinical scenario, you might want to optimize for one or the other. So here on the right, we have a plot of all the possible sensitivity and specificity values for MAL-ID for distinguishing lupus from the other diseases in this model. For example, we can achieve 93% sensitivity and 90% specificity. And that's an exciting result, we think, because today's lupus tests are often highly sensitive or highly specific, and usually not both. So this may be the beginning of a new biomarker approach, although it, of course, requires further validation. And at the moment, it's limited only to distinguishing lupus from the other diseases in this particular data set. While these results and these scores are exciting, one of the most important things we try to evaluate in our study here is, are we getting fooled by the machine learning? Are we actually picking up on real biological signals, things that are genuinely associated with these diseases? Or are we picking up on some sort of hidden differences between the patient groups that come from how we've constructed our data set? This is a common problem in machine learning, right? Is your model latching onto the real thing, or is there some sort of confounding signal that might not be obvious to us, but is going to be what the model latches onto and prevents it from working in real life scenarios? A classic example from the machine learning literature here is a dog versus wolf classifier. You might think that your model is able to tell dogs apart from wolves in photos really well, but it turns out that all the wolf photos happen to have snow in the background. So if you look at what the model is actually picking up on, it's picking up on the snow rather than something about the animal itself. And the consequence of that is that the model might not work in real life with a new dog or wolf photo necessarily. So we set out to see what our model might be latching onto. And this was a big part of our project. We're just going to give a quick summary here of a few of the ways that we've tried to do this. The checks included checking how big a role batch effects played in the results. Does the model work on external independent data sets generated by other individuals? What is the demographic composition of our data set, and is that driving the results in any way? And then peeling back the layers of the onion of how our model works, could we tell which B and T cells in particular are driving the predictions for each disease and see if they make sense to us based on what we already know in the literature about these conditions? So the first focus was on batch effects. This is the concern that what you're learning is not the disease biology, but rather a noise or technical signal from the way you process the samples in the lab. So here's a hypothetical extreme example of potential batch effects. Imagine you sequenced all of your COVID-19 patients on a Monday, all your healthy controls on a Tuesday, and all of your lupus patients on a Wednesday. And then your model seems to be able to distinguish these three categories, let's say. Again, this is just a hypothetical. Our data set was not constructed in this way. But when you see that the model is able to distinguish the categories, how do you tell, is it picking up on something genuinely related to the immune response to these diseases? Or is it just learning Monday versus Tuesday versus Wednesday because of some sort of hidden differences in how your sequencing operation ran on those days? This is a very tricky thing to evaluate. So here's a few ways we approached it. The first was to test if our model is actually learning some real signal. We added another batch. We added a new cohort of COVID-19 patients, just to follow through this hypothetical. Imagine it's on a Thursday that you get this new group of COVID-19 patients that you sequence, and now you can train your model on the data you have until then, excluding that new batch, excluding that new cohort of individuals, and use those as a held out set, as if they were new patients walking in the door. And if the model gets them right, then that's a good signal that it's learning something real about the biology, something actually COVID specific that is generalizable to new cohorts, as opposed to a batch specific pattern that we might not see in new data at all. In this case, what we saw was that when we did this experiment with a new COVID-19 cohort, all of the patients there were correctly classified. We also did similar tests with lupus patients and healthy controls, and we saw that the results were consistent with the overall accuracy of our model. So this increases the likelihood that what our models are picking up on are true biological signals rather than batch effects. And next, we took this a step forward by also checking external data sets generated from other labs who, in case there's a remaining concern that there's something specific we're doing in how we're constructing these data sets, let's go and test these on other data in the wild. And during the pandemic, there were other B-cell and T-cell receptor data sets published from COVID-19 patients and from healthy controls with similar sequencing protocols. So we trained a model on all of our data and then evaluated it on this external data that had never been seen in the training process. And again, we're seeing high performance on this external held out data for B and T-cell receptor based prediction of are these COVID patients or healthy controls. So those data points are intended to show how we came around to believing that what we're actually picking up on are the real biological signals as opposed to certain batch effects. But we did a few other checks as well. If we believe that batch effects are minimal, the next thing we asked are there demographic effects? And there are numerous ways that subtle demographic-based differences can manifest in a patient's B and T cell receptor populations. So just to give two examples, a person's age and where they live can influence the prevalence of disease and the environmental exposures that they may have had, which shape their immune repertoires. And ancestry can influence the genetic building blocks that are available in an individual to construct those VDJ rearrangement sequences that Scott was describing earlier. In fact, our cohorts do have different demographics, so we had to check how much could this explain the ability to distinguish diseases in this data set. On the left side here, what we tried was to create a model that doesn't use sequence information at all. It only uses age, sex, and ancestry to predict disease. And what we see there is a moderate score of 0.85 AUC, meaning this model is picking up on the subtle attributes of the demographic makeup of our different patient groups. And what you'll notice in particular is it's getting the HIV patients correct. It's getting those predictions right. And that's primarily because our HIV cohort is an African cohort living in Africa. So the model here may be learning to predict that anyone with African ancestry has HIV, which is obviously incorrect. So this demographics-based model could make mistakes for other people who are not HIV patients. And now you can compare that to the model on the right, where we have the sequence information and the demographic information available to the model. So we gave it everything we have. And this model that can see all the information available achieves 0.98 AUC. The takeaway here is that demographic information is not enough. It's the sequence information that gets us to a diagnostically useful level. I will hand it back to Scott now to discuss how we started unraveling what the model may actually be picking up on as far as which sequences matter.
Okay, yeah, thank you, Maxim. So, and as is clear from this, right, this initial set of samples and conditions, this is not the actual diagnostic dilemma that anybody's going to face in the clinic, but they were selected to be basically initial conditions that we should be able to tell apart if there is this sort of information in the immune repertoires. So that's something that we want to be careful about. And if you look at the paper, it's about 1/3 explaining the model and showing our results, and 2/3 trying to go through all these different ways that we could be fooled, or ways that the machine learning method could be actually picking up on false signals. So we also wanted to look and see whether the sequences that seemed the most predictive for particular conditions matched anything that we knew from prior immunological literature or any other data that were available beforehand. So we looked, for example, if you look at the different V genes that were prioritized in the model for predicting the different disease categories, did they match up with things that we knew about in advance? So, for example, that was really true for COVID-19. You can see that some of the most predictive ones, for example, this V124 category, that's a really heavily overused variable gene segment in the heavy chain of antibodies that recognize the N-terminal domain of the SARS-CoV-2 spike protein. So this actually matched really nicely with the ones that we expected we should see, and they showed up in our analysis. Similarly, there's other sort of common public sequences that use V434, V439. And we also noticed that in different conditions, like for example, in the lupus classification, the model made a lot more use of IgA sequences than just the IgM and IgG, which also I think is correlated with detection of unusual IgA expressing B cells in lupus as well. We also saw for flu, for example, flu vaccine response, some really well-known public sequences that showed up in that as well. So that sort of was reassuring, because the model knew nothing about public sequences, had no input from external data, otherwise from these public things that were already known from previous immunological research. Let's see. And then, yeah, I think just, ways that we're trying to extend this now, or trying to apply it to other conditions. We're applying it to a lot more disease subtypes and conditions, especially autoimmune diseases, but many other categories of human immunological exposures. And we really want to try to focus it on things that would actually be real diagnostic dilemmas, or things that would be useful, to make a distinction where the current diagnostic tests are not that great at distinguishing what condition somebody has, maybe especially early on, if they haven't fully, declared what the phenotype is going to be in terms of the progression of the disease. We want to improve ways that we can quantify the degree of uncertainty in the classification. And so, that goes into deciding whether we're going to have a test that would look for one condition in particular, or distinguish a particular set of conditions from each other. Also to be able to evaluate, the effect of other things like comorbidities, right? Patients don't show up with one pure illness a lot of the time. They might have several things going on. So we want to see if we can detect multiple different things happening in a particular individual. And that's something that also, for all of its harms, like the COVID pandemic was pretty helpful for that, because if we have samples collected before 2019 for a particular disease state, and ones with the same other disease state, like let's say an autoimmune condition pre and post COVID, in the ones post COVID, we see two signals. We see that they have COVID-19 associated sequences, but they also have the ones associated with the main disease that we are interested in. So that's sort of a reality of how the immune system is keeping a memory or a record of the previous exposures. One thing that we didn't spend the time on today, but is we noticed that reassuringly, when we looked, for example, at the lupus patients that the model got wrong, they often tended to be the ones that had the lowest clinical disease scores. There's this sort of SLEDAI score for saying disease acuity or disease activity. The ones that the model thought were healthy were actually the ones that had the lowest SLEDAI scores. So that actually is telling us that probably we can tell degrees of severity of diseases as well with this potentially. We also, we think that eventually it may be possible to detect new conditions that haven't been encountered in the sort of modeling, but that's something that will be for future work. Also, it's clear for certain kinds of conditions, like an acute infection, where you want to find out what's the patient's illness, what are they infected with. This would take some time, this particular approach, because you have to amplify the sequences from the blood, you have to sequence them, you have to analyze them. So certainly for most infectious disease conditions, this may not be the most immediately useful approach to use, because it might not be fast enough, right, compared to other methods of detecting a pathogen. So yeah, that's basically, but the advantages are you basically can collect one blood sample and diagnose any condition from it potentially. And it's sort of a relatively easily available sample, just our usual blood sample. As we've shown, it does seem to do a pretty good job in distinguishing the conditions that we trained it on in this first set of data. We need to look at a lot more people, many more conditions to really further flesh that out. And this is really one of the first efforts in the literature to make use of both the B cell and T cell arms of the immune system in one sort of combined classification state. Immunologists tend to be sort of specialized in one cell type or the other, and even subspecialized in particular subsets of those. Immunologists are great splitters. So, but this is actually trying to make use of as much information as we can. It could, of course, be combined with other kinds of measures, like overall gene expression from non-lymphocytes, or secreted proteins, or serology. So anyway, with that, we'd like to thank all of our collaborators, and especially Anshul, Rob Tutroni, and Aaron, who are the other main players in the study, but also our many other lab members and collaborators, a number of whom are at Stanford and we're really appreciative of, but also across other medical centers. And most of all, we thank the patients who contributed their biosamples to this work. So thanks for your attention, and we're planning to have a discussion now, I think, with Dr. Bhatt, and you guys can ask questions and sort of further grill us on this. So, yeah, thank you.
Wonderful. That was incredible. I think I got asked to sort of help talk about some of the clinical applications of this model. And one of the things I remember from my training, which admittedly was back when dinosaurs walked the earth, I used to love coming to grand rounds because we got to talk to these incredible scientists about their work and then really start thinking about, well, how can we apply this to what we do as clinicians? So I was hoping we could take a little time to do that. And the first thing I sort of wanted to discuss is this is a whole new model, a new way of looking at disease. And I thought it might be helpful to put in context a little bit, what are some similar types of new approaches we've used that have over time become common in diagnostics, so sort of molecular genetics type things.
Yeah, so one kind of parallel would be for cancer diagnostics, over the past 10 or 15 years, once high throughput DNA sequencing became available, it's become clear that, different cancers sometimes share, similar gene mutations or actually have some common genetic features that were just not appreciated before. And within a category of cancer, finding out which mutations a particular cancer has can be helpful in predicting the prognosis to particular treatment, response to particular treatments, the patient prognosis from that. I think that there's some parallel to the really diverse immune repertoires where particular sequences amongst that whole, diverse set of rearrangements can actually be pretty informative indicators of what immune response is going on. And, in the case, maybe what autoimmune reaction is happening in the person's body, things that are really difficult to study otherwise. So I think that, as we continue sequencing more and more patients with these kind of data, I think we'll be able to identify those shared sequences, even if they're relatively rare, between pairs of individuals or, larger numbers that have those. And something similar, like, in infectious diseases, being able to sequence, viral sequences, or at least detect nucleic acids, has been a much improved way of identifying what the cause of someone's disease state is. So I think it's just lagged a bit behind in the immunological world because these data are just by their nature very complex and require a lot of individuals to be examined to really detect these common patterns. That's, I think, something in that direction.
I think if I may just add one more thing, one of the through lines there, I think, is that in the infectious disease example, we rely on direct detection of the pathogen as the gold standard. In cancer, the driver mutations. And here, perhaps in the autoimmune world, this may be a window into the specific cells that could be driving the autoimmune activity. Of course, this requires further validation and testing, but if we do have this way of finally drawing insights out of these very complicated data sets of the millions of immune cells in a person's blood sample, perhaps there could be that more mechanistic look at what is actually fueling the fire.
That's fantastic. And that sort of draws a parallel I'd like to bring out a little bit more about really sort of how is machine learning and AI involved in this? Because we think about AI, we think about large language models, we think about ways of helping give us responses to patient questions. How is the type of machine learning that you used in Malady different from a large language model? I know you talked about that a little bit in your speech, but I'd love to just have you draw out a little bit more of the differences and how that will affect its utilization as a clinical diagnostic.
Sure. Do you want to tackle that?
Yeah, I can start. I think when we think of large language models today, or ChatGPT or something like that, those are often black boxes that have been fed a lot of unstructured data of, here's the vast extent of the human language out there, and they learn how to fill in the blank and how to generate new text or new images, and often have hallucination problems as a result. This approach is a bit different because while we are using similar building blocks to extract features that are meaningful out of these biological sequences, we then do train the models with gold standard data, with information that is very structured about what is this patient, how have they been diagnosed, what is their disease activity level, or other factors like that have been carefully annotated by physicians. So there's, I think, a fundamental difference there between the black box approaches that are just fed everything in an unstructured way versus this very carefully supervised approach that also has been designed to allow us to peel back the layers a bit to understand, is it picking up on real signals, or are we being fooled in some way by the machine learning?
So basically, you're giving the model information about this is what might be biologically relevant. Can you find these signals and pull them out and help distinguish one group from another?
I think that captures it. That said, though, we still need to be very thoughtful if this one day is being used in the clinic, perhaps down the road. There's a lot of very important questions to address around AI fairness and making sure that this model is not getting fooled in ways that we haven't been able to detect already. And so that'll require a lot of very diligent work. Today, this is a proof of concept study, and I want to make sure to emphasize that.
Absolutely understood.
Scott, is there anything you want to add?
Yeah, maybe related to that, it's also possible, I think, that conditions that are currently diagnosed as one thing, right? Many of them may contain subgroups or subcategories. This way of looking at the different immunological conditions might actually be able to tease out, the different subgroups or maybe things that would have importance in terms of how you might want to treat a patient, or, whether they're going to have rapidly advancing disease or, indolent course or something like that. So I think that's also, our current gold standards for some conditions are not all that golden, and maybe this could help to improve diagnostic work overall. But that would require a lot of follow-up or seeing how people respond in trials of therapeutics and things, of course.
That's a great segue into some of the questions I sort of how we as clinicians might think about where will we use this eventually. And I am a primary care physician. And so for me, I often am struck with patients with first presentations of syndromes. So, for example, a patient comes in with fatigue, some joint pain, sort of a not well-defined syndrome. And one of the first questions I often will ask is this a rheumatologic condition or not? Is this the type of situation where Melody might eventually be useful? And how would you think about employing that?
Yeah, I mean, I think it could serve in that role of helping to narrow down what overall category of disease somebody has. That remains to be validated. I think in our current results we can't make that claim, but it seemed like one possibility for this could be that, for example, if you have a patient presenting in a primary care clinic, maybe in a region of the country or in a region of the world where it might be very difficult to get a referral to a rheumatologist, quickly or something, maybe this would be better than some of the first line testing that would otherwise be available, like ANA antibody test or something like that. And maybe it could actually even provide more precise diagnostic information at an early stage that would help to direct patients where they could, get care that's appropriate to their condition more quickly, or else at least prioritize patients that need to be seen right away because they're going to have a rapidly advancing destructive condition. But that's just sort of hopeful speculation about how this may play out in the longer term. But, Maxim, I don't know if you had thoughts on that.
And as we've been trying to think through where this may fit in down the road in the diagnostic arsenal that clinicians have access to, another topic has been a little bit past the primary care time when you are now being cared for by a specialist, but there's still uncertainty about which specific diagnosis you have. A lot of these diagnoses, as we've come to understand, are clinical diagnoses. And if we could provide additional biomarkers to complement today's workups, maybe that could be useful. Again, as Scott very rightly pointed out, that all requires additional validation and development, and we're nowhere close to that point. So I want to make sure that's absolutely clear. But a big part of our thinking right now is trying to figure out where would this be useful, because these workups are already so complicated. How do we make sure that this is useful information that supplements what is already available?
And then we didn't get it all into the regulatory aspects, right? I think the FDA likes tests that are very cut and dried, testing for one analyte, being able to really show, really convincingly that you can detect that thing sensitively and precisely and repeatedly over time with no drift in the results. So these kind of AI analysis approaches being applied to complex data sets like BCR, TCR sequences are a more difficult area to actually, regulate and to have good quality control in terms of how the assay would be done. So that's also work that needs to happen.
One of the areas that I know can be very difficult diagnostically are central nervous system conditions. We're often with things like multiple sclerosis, we know that, or we suspect there's an autoimmune component, but we're left with imaging and clinical progression as one of our ways of diagnosis. Could you see a way where this type of testing could be used to sort of figure out, is this MS or not MS? Because that's certainly a question I get asked by patients all the time. Is there a way you could see something like that happening over time?
We hope so. We have not tested that, just to be clear, but we hope that based on the promising results with this original proof of concept study, that we could extend it to additional autoimmune diseases. I think with MS in particular, there are a number of really exciting questions here beyond just the diagnosis as well. Once you've been treated, have we actually tamped down on the disease-specific immune activity? That appears to be quite hard to understand and measure today. And if we actually are finding the disease-associated immune cells that may be pathogenic, perhaps this could be a different way to address that question. But all that needs further study. We haven't tested that yet.
And that brings to mind something you brought up in your talk about the lupus patients, where when your model made some errors, it appeared to be in the patients who had milder disease. Could this be a model over time that could be used to predict how patients are responding to medications? For example, I know in the rheumatologic conditions, it's often trial and error. And would this perhaps be a way to find out earlier, are patients responding, and can we use it to direct therapy?
Yeah, again, I think so. I mean, it looked like in the lupus case, we definitely, if you had milder disease, it showed up as a less strong signal in the initial classification. So we guess that response to therapy might show up as a similar decrease in the disease-associated features, but that's also stuff we need to really validate with larger data sets with carefully controlled trial conditions and time points to make sure that we're looking at that carefully. But that's certainly, I think, reasonable, and especially if you think of more extreme treatment options like CAR T cell approaches being used to treat autoimmune diseases now, and in initial testing, being able to tell whether the disease signatures that were characteristic of the person's disease state before they had that treatment, whether that's been eliminated by the CAR T treatment, would be a good thing to look for, and maybe it might be able to predict if the disease is going to recur. Some of the same cell clones have actually survived that treatment and might be causing the disease to come back. Then that may be something that's good to detect, but that's all, again, still speculation beyond what we have data for right now.
Of course. And I think this goes back to what Scott was saying a bit earlier with trying to use these models to distinguish subtypes of these diseases. Today, we think about, in this work, we've thought about just the large disease categories as a whole, but perhaps we could train these same models to predict, who would benefit from a particular drug, let's say a TNF inhibitor, right? And see if there are certain relevant subtypes in there that could be used to reduce the trial and error and reduce the pain and suffering that goes into it.
And in some of these diseases, I see a question from the audience.
Yeah, great.
Mike. Thanks. Did you sequence MHC and HLA, and did those data enhance your model, or is your model independent of those data?
Yeah, that's a great question. We didn't sequence the MHC in these individuals at all. So that would be an area for potential improvement, because we think that the TCR sequences in combination with the MHC may be even more informative. But for the purpose of this, we were just sticking to the receptors themselves. We think that's also an important aspect of why patient genetic ancestry shows up as a feature that is partially predictive of somebody's condition. Is the TCR repertoire is definitely shaped by what MHC alleles you have in your genome, and so those two are linked. And that's also just sort of an important overall caveat for this is, patient demographics affect your immune repertoires, right? But that also is associated with the just prevalence of different diseases in different groups. So it's we're not ever going to be able to completely untangle those things, but we need to be able to sort them out and make sure that we're not getting fooled by a signal which is just a broader demographic one associated with that condition, as opposed to the disease-specific immune receptor features. Yeah, so thanks for that's a very good question.
First of all, thanks for coming to Grand Rounds, and congratulations on the incredible work. As I hope there are 100, 200 people online that are part of this, and we are hopefully bringing people back to Grand Rounds in person soon to fill up all these empty seats. Anyway, I really appreciate it. As you know, I've been following your work for a while, and I think, this has to be so much the future of medicine. And what we've seen, I think, in genomics over the course of the last decade is going to be the next decade, I think, is immunomics and moving towards this. The ankylosing spondylitis paper you showed, I think, is a really interesting example of the potential essentially for clonal ablation and thinking about autoimmune disease in a very specific way. So two more practical questions, though. One, how much does it cost now? Where are we on the cost curve for one of these tests? And what is step 2, 3, and 4 to get to that world where anyone who wants it, who comes into a primary care office, could have this test and have an early diagnosis leading to a precision therapy?
Yeah,
Easy question.
maybe a starting, so starting one would be, cost-wise, the sequencing that goes into this sort of analysis, it's way less than you would need for a whole genome, like clinical genome sort of sequencing. So from that perspective, the actual material costs of doing the sequencing are probably less than $100 per patient. Probably could be way less depending on exactly how you did it. And I think, some of the main steps toward actually applying it for real clinical cases would just be, we need to look at many more individuals with well-characterized clinical phenotypes and determine what those patterns are for them. I think, one question that comes up is like, how many patients do you need to look at to detect a reasonable signature that lets you identify that condition? And in our testing so far, it's somewhere like 30 to 50 is about the number of patients that we found gives you a pretty good signature for a particular disease state. But we think that for this to really perform well and be broadly useful, we want to be looking at, training numbers of patients in the hundreds or essentially thousands to really be able to tease out things that are truly disease specific. So that's stuff that will require a scaling up of the number of individuals that we look at, collaboration with a bunch of the other medical centers that have large, well-phenotyped cohorts of patients. And and I think really as Maxim pointed out in part of his section of the talk, important question is like, what are you trying to distinguish from what else, right? Maybe this could be a universal diagnostic, but I think it's going to be more easily applied to cases where there's a specific diagnostic question for a category of diseases and you want to narrow it down, or you want to at least, identify whether somebody is in a broad group of like, autoimmune versus infectious, like encephalitis versus like, other immunological attacks on the nervous system, for example. So I think those are some points relevant to your question, but Maxime, do you have any other thoughts?
No, I think you covered it.
Yeah.
I think if you guys don't mind, we have a bunch of questions both online and in the room. Maybe we'll just do a couple more, if that's okay with you. We'll go a little bit beyond and be respectful of people's.
Thank you so much for the presentation. You don't need more variables to look at because you've had plenty. However, one of the very many places that we don't do well is recognizing the different genetic predispositions in ethnic groups and socio-demographic groups. Did your data help you to help differentiate gender, ethnicity, socio-demographic data as you looked at the predisposition to these illnesses?
Yeah. Do you want to address that, Maxime? Or
Uh,
I can say a few
please.
comments to make. So, I mean, we wanted to look at that general topic area of potential confounders carefully. One of the things we tried to see is, how well can we actually detect the demographics of patients based on the immune receptor sequence data themselves, right? Can you tell if somebody's, from a European, Caucasian background compared to somebody who's, recent, African individual living in Africa, that sort of thing? And the answer to that was, we can tell those differences, reasonably well, not as well as the actual disease-specific signatures that were the main purpose of the overall exercise. We couldn't tell male from female very well at all, which I thought was interesting because, for autoimmune diseases, of course, some of them are quite enriched in females compared to males, and a few are male prevalent. But age, you can tell somebody's age roughly with this sort of approach, but not very precisely. So I think that there's, the demographic main variables we would think about initially, they have some imprint on the data that we get with the receptors, but they don't dominate the patterns overall. And one example of that was amongst the lupus patients, we had pediatric lupus cases as well as adult, and they all seem to be classified reasonably well as lupus, as opposed to those other big differences between pediatric immune repertoires and adult ones. So we think that the disease-specific signatures are pretty strong. At least for the ones we use for this paper, which were mostly the conditions where we had enough individuals to really try to do this. Yeah, so it's a very important direction, and we know, like, autoimmune disease presenting in a particular patient demographic population can show up somewhat differently, or, everything, as we've seen in lots of other areas of medicine, like, different gene mutations can contribute to a similar phenotype in like, inherited disorders, for example, but that's stuff that we still have to explore in a lot more depth with these immunological classifications.
think we'll do one more question here from the room, Doctor.
Thank you so much for your fascinating work. You already mentioned your future direction to try to distinguish patients with comorbid conditions, and I was particularly thinking about people with a chronic illness like lupus or multiple sclerosis in the future who has an acute SARS infection. But my question is whether or not your model will be useful in distinguishing disease progression. So we know that we have people who are chronically stable and then they hit an inflection point where they acutely decompensate. And specifically, could this be useful in detecting, say, a rejection in cellular or antibody-mediated rejection in patients post-solid organ transplant, or myocarditis in patients with checkpoint inhibitors?
Yeah, we definitely think that transplant rejection is an area where you might see a big signature, of the T cells that happen to be responding to some, mismatched allele or minor antigens that are mismatched between the transplant recipient and the donor. That's something we haven't explored yet with real data in our studies, but similarly with the autoimmune side effects of checkpoint blockade treatments, that's something where we certainly think those are tissue specific, and those should be associated with characteristic receptor sequences. So that's an area we're also interested in, and we would expect that it should have some value there. But again, this is pre-data, speculation. Yeah.
There's evidence to suggest that with regards to the tissue-specific complications, perhaps, of autoimmune disease, just to take that example, there's evidence to suggest that there's recirculation of immune cells from those tissues into the blood. We still have to evaluate how strong that evidence is and whether that is something we can pick up in our data, but that lends us some hope that you could apply this sort of technique for these much more granular monitoring tasks.
Yeah, there's some other unusual situations, like, it's been shown in just recent years that if somebody gets a measles infection, wipe out a lot of their immune memory for other conditions, right? So that's something that, could show up as a signal of an absence of, signals that would have been there otherwise. I never thought we would have been talking about that as a realistic health concern in the US in 2025, but that's actually, there's a lot of other ways that the immune system could inform us about what's been happening with an individual.
I was hoping to end on a happy note. That wasn't the happy note.
That's right.
But the science is indeed so optimistic, and I have a good sense that we will have you back to talk more. I'm sorry we didn't get to all the online questions. There are many participants online, and we will share those questions with you. Please come back and join us. Thank you all for joining in the room and joining online.
Thank you all.
Thank you for the question. Thank you, Dr. Bhatt.
Thank you.
Yes.
The preceding program is copyrighted by the Board of Trustees of the Leland Stanford Junior University. Please visit us at med.stanford.edu.